Good morning, everyone. I am Dr. Mayuri Rani, and I am delighted to welcome you all today. Today, we will be exploring an exciting topic: conventional versus molecular microbiology, debunking myths. We'll take a closer look at the differences between these two approaches, clear up some common misconceptions, and understand how advancements are shaping the future of microbiology. So, let's dive in and uncover the truths behind these fascinating methods. Let's take a closer look at the key differences between conventional and molecular methods. Traditional, or conventional, methods usually focus on observing the biochemical characteristics or physical traits of pathogenic organisms. This means we look at things like their appearance, behavior, or how they react in certain tests. These methods have been used for a long time and are quite reliable, but they can sometimes be slower or less specific. On the other hand, molecular methods take a different approach. Instead of looking at physical traits, they target specific parts of the organism's genetic material, its genome, or other molecular markers that are associated with pathogens. This allows for much more precise identification and often faster results. Understanding these differences is important because it highlights how advances in molecular biology are transforming how we detect and identify pathogens. This shift is central to improving diagnostic accuracy and speed, which ultimately enhances our ability to respond effectively to infectious diseases. So, let's talk about the benefits of conventional microbiology culture compared to molecular methods. With conventional culturing, the approach isn't targeted at specific organisms. Instead, it allows us to grow and identify a much wider variety of pathogens. This can be particularly useful because there are many organisms that might not be included on a standard molecular panel. For example, I once encountered a case where a BioFire JI panel test came back negative. However, when we did the culture, we actually isolated S. epidermidis. This highlights how conventional culturing can sometimes detect organisms that molecular panels might miss, giving us a broader picture of what's really going on. Overall, the main point is that conventional methods offer a more comprehensive view, especially when we're unsure what pathogen might be involved. It's a valuable tool in our diagnostic arsenal, particularly for uncovering unexpected or hard-to-detect organisms. One additional benefit of the conventional culture approach is that it allows us to quantify organisms, which is something most molecular panels currently cannot do. For example, in urine cultures, we can determine the actual number of bacteria present, giving us valuable information for diagnosis and treatment decisions. Culture also forms the foundation for producing antigens used in serological tests, both in the lab and in live disease models. This makes culture irreplaceable when studying emerging pathogens, as it enables us to conduct further genetic research to understand how these microbes cause disease. And once we identify the organism, we can also perform susceptibility testing across a broad range of antibiotics. This helps us guide effective treatment options, ensuring patients receive the most appropriate therapy based on the organism's resistance profile. So overall, culture remains a critical tool in microbiology, offering unique insights that complement molecular methods and enhance our understanding and management of infectious diseases. Now, continuing our discussion on the benefits of conventional methods over molecular ASD from this perspective, let's delve deeper. Although modern genotypic methods seem promising, they offer rapid and potentially more accurate detection of resistance determinants. There are still important reasons why phenotypic methods remain crucial. First, resistance in microbes can be driven by multiple mechanisms. This means that a single resistance trait might involve various genetic factors. The problem is, the genetic landscape is often so diverse that current molecular techniques might not capture all these mechanisms effectively. In other words, the genetic basis of resistance can be quite complex and may even surpass the diagnostic capacity of the molecular tests we currently have. Second, it's essential to remember that while molecular methods can predict resistance, meaning they can tell us if resistance genes are present, they can't guarantee how the pathogen will actually respond to the drug. In other words, the presence of a gene doesn't always mean the organism is resistant in practice. And finally, we have to consider the practical aspects. Molecular techniques tend to be more expensive, and they are sensitive to contamination, like carryover of genetic materials. That can lead to inaccurate results. So, despite their advantages, these limitations mean phenotypic methods will continue to play a vital role in antimicrobial susceptibility testing. Overall, it's clear that both methods have their strengths, but understanding their limitations helps us make better clinical decisions. Now, moving on to some of the limitations of molecular ASD from a conventional perspective. Thirdly, sometimes the normal flora present in a clinical sample can also carry resistance genes. This means that molecular methods, which detect these genes, might not always give us a clear picture. Their results can be tricky to interpret correctly because they don't distinguish whether the resistance genes are in the pathogen or just part of the normal flora. Additionally, there are practical situations where traditional culture-based methods are still indispensable. For example, determining the minimum inhibitory concentration, or MIC, of an antimicrobial against a specific isolate is critical for guiding therapy. Also, preparing a hospital antibiogram, which helps us understand local resistance patterns, relies heavily on phenotypic, culture-based data. These methods are foundational in modern infectious disease management, ensuring we have accurate and actionable information to treat our patients effectively. What are the benefits of conventional methods like microscopy? Let's continue exploring that. A common myth today is that microscopy is an outdated technique and that it's no longer useful. But the truth is, microscopy is actually the oldest diagnostic method we have. It's simple, reliable, and still very cost-effective. In a bacteriology lab, for example, gram staining followed by microscopy is typically the first step we take when examining clinical samples. This initial test helps us decide whether further testing is necessary. It often reduces unnecessary processing of specimens, and importantly, it increases our chances of detecting the pathogen quickly and accurately. This not only saves time, but also reduces costs for the laboratory. Additionally, we have grading systems like Murray and Washington criteria or Bartlett's grading system. These tools help us differentiate between bacteria that are just colonizing or harmless and those that are true pathogens causing disease. By applying these standardized methods, we improve our diagnostic accuracy, which is crucial for effective treatment. So, despite the advances in molecular techniques, microscopy remains a fundamental, practical, and valuable tool in microbiology. It connects to the overall theme of emphasizing the importance of traditional methods in modern diagnostics. Now, let's explore the benefits of conventional methods like microscopy. Firstly, the traditional zeal. Nielsen staining remains a cornerstone for the early diagnosis and management of tuberculosis, especially in resource-limited settings where advanced techniques may not be available. In remote areas lacking facilities for Mycobacterium tuberculosis culture, sputum smear microscopy becomes even more critical. It serves as a key indicator for predicting how well a patient is responding to antitubercular treatment, particularly among those who are bacteriologically confirmed. Moving beyond TB, microscopy also plays an essential role in diagnosing other infections. For example, detecting gram-negative bacilli or budding yeast cells in cerebrospinal fluid or blood can provide life-saving information. These simple microscopic observations can guide timely treatment decisions, which is crucial in critical cases. During the 2021 mucormycosis outbreak in India, a straightforward technique using light microscopy with potassium hydroxide proved highly effective. It allowed us to identify broad, aseptate hyphae in about 70% of suspected cases during the second wave of COVID-19. So, despite the advent of newer technologies, conventional microscopy remains invaluable, particularly in settings with limited resources. helping us diagnose and manage serious infections effectively. Now, let's continue exploring the benefits of conventional methods like microscopy. As we've seen, microscopy plays a crucial role in diagnosing parasitic infections. Its importance cannot be overstated, especially when we consider the challenges associated with other diagnostic techniques. Culturing pathogenic parasites, for example, is very labor-intensive, requiring meticulous attention and strict biosafety measures. Molecular techniques, on the other hand, although highly accurate, tend to be costly and demand advanced technology and expertise. That's why microscopy remains the go-to method in many settings. It allows us to detect parasites like Plasmodium species and microfilaria directly in peripheral blood smears. Similarly, it's essential for identifying amoebae and helminths in stool wet mount preparations. So, despite advances in molecular diagnostics, microscopy continues to be vital because of its practicality, accessibility, and reliability in routine parasitology diagnosis. Let's explore the benefits of conventional methods like serology. Some investigators argue that serological tests primarily aim to demonstrate the patient's immune response to an invading pathogen. In other words, they provide indirect evidence of infection, which can vary quite a bit depending on an individual's genetic and immunological makeup. This means that results can sometimes be inconsistent or hard to interpret. These tests also face challenges due to cross-reactions and antigenic similarities between different bacterial genera, making it tricky to pinpoint exactly which pathogen is responsible. Now, on the flip side, molecular tests such as PCR offer high sensitivity and specificity, especially for diagnosing many infections, even those caused by less common or unusual pathogens. But serological tests still have their place. For example, by tracking seroconversion, meaning the rise or fall of antibody levels, they can give us valuable insight into the natural history of a disease. And with the advent of newer tests, like the enzyme-linked immunosorbent assay, we can differentiate between immunoglobulin M and immunoglobulin G antibodies. This distinction helps us determine whether an infection is recent or has occurred in the past. So, overall, while molecular methods are powerful, serology remains a useful tool, especially when it comes to understanding the progression and timing of infections. When we compare molecular methods to conventional ones, the first thing to highlight is their remarkable sensitivity and specificity. Because they amplify and detect specific genetic material, they can identify even tiny amounts of pathogen DNA or RNA, making them much more accurate. Another significant advantage is speed. Molecular testing can often deliver results days earlier than traditional culture methods, which can take weeks in some cases. This faster turnaround can be crucial for timely clinical decisions. Additionally, many molecular tests require less hands-on time in the lab. This means they can streamline workflows, reduce workload, and minimize the chance of human error. All these factors, improved accuracy, faster results, and efficiency, can have a profound impact on patient care. They help clinicians make quicker, better-informed decisions, which ultimately improves patient satisfaction. From a broader perspective, these benefits also affect healthcare systems by enhancing workflow efficiency and potentially reducing costs. So, in summary, molecular methods offer a powerful advantage over conventional techniques across multiple facets of clinical practice and healthcare management. Now, let's talk about some additional benefits of molecular methods compared to conventional ones. One of the key advantages is that molecular techniques can analyze a single panel, like the GI panel from BioFire, to detect bacteria, viruses, and parasites all at once. This means we get a comprehensive picture quickly, rather than running multiple separate tests. Another important point is that molecular methods can identify atypical bacteria that are difficult to grow using routine cultures. For example, with the pneumonia panel, we can detect organisms like Chlamydia pneumoniae, Legionella pneumophila, and Mycoplasma pneumoniae. These are often tricky because they don't grow well in traditional cultures, but molecular testing can identify them directly from samples. Lastly, for sexually transmitted infections, molecular tests like the ctNG GenExpert allow us to detect Chlamydia trachomatis and Neisseria gonorrhoeae rapidly and accurately. This quick detection is crucial for prompt treatment and preventing complications. Overall, these molecular approaches enhance our diagnostic accuracy and speed, which ties into the larger theme of improving patient care through advanced testing methods. Let's continue exploring the benefits of molecular methods over conventional ones. Quantitative PCR for HCV and HBV is particularly valuable because it not only helps us detect the presence of the virus, but also allows us to monitor how well patients are responding to treatment. This means we can get a clearer picture of prognosis and adjust treatment plans accordingly. Additionally, tools like GeneXpert and line probe assays are game changers because they can quickly identify resistance to first and second-line drugs. This rapid detection is crucial because it helps us decide on the most effective antitubercular therapy and determine the optimal duration of treatment. The speed of these tests is especially important. It enables us to start treatment early, which can significantly prevent further complications, reduce morbidity and mortality, and also curb the spread of infection within the community. Overall, these molecular techniques enhance our ability to deliver precise, timely, and effective care, aligning perfectly with the overarching goal of improving patient outcomes. So, let's talk about some of the limitations of molecular methods compared to traditional approaches. One key point is that molecular techniques rely on using specific primers, which means they target particular pathogens or markers for antimicrobial resistance. While this can be very precise, it also means that they might miss organisms that aren't specifically targeted. Another important aspect is that these methods can't distinguish between viable and non-viable organisms. So, you might detect DNA from dead bacteria, which doesn't necessarily indicate an active infection. Cost is also a consideration. Molecular methods tend to be more expensive than conventional tests. And, as we've seen earlier in the presentation, they usually don't report the quantities of organisms present, which can be important for understanding the severity or stage of an infection. Overall, while molecular techniques are powerful, these limitations highlight why they are often used alongside traditional methods to get a more comprehensive picture. So, when we compare conventional microbiology methods to molecular techniques, there are some clear limitations to keep in mind. First, conventional methods tend to have lower sensitivity and specificity. This means they might miss some infections or incorrectly identify others, which can impact patient care. Additionally, the turnaround time is generally longer. It takes more time to get both identification and susceptibility results using traditional methods, which can delay treatment decisions. Another important point is that conventional methods depend heavily on the viability of the organisms. Some bacteria or fungi are really difficult to cultivate in the lab, making detection quite challenging. Finally, these methods also require a certain level of expertise to interpret cultures correctly. Reading and understanding culture results isn't always straightforward, and it depends on the skill and experience of the laboratory personnel. Overall, while conventional microbiology has been a mainstay for a long time, these limitations highlight why molecular methods are becoming increasingly valuable in rapid and accurate pathogen detection. So, let's talk about some of the common conventional methods that have been replaced by molecular techniques in laboratories. These traditional methods, like culture, used to be the go-to for identifying pathogens, but now molecular methods have taken their place because they're faster and often more accurate. For example, instead of waiting days to grow bacteria in culture, we now use things like gastrointestinal pathogen panels, which can quickly detect multiple pathogens from a single sample. The same goes for meningitis and encephalitis panels. These allow rapid diagnosis of serious central nervous system infections. Other examples include tests for Streptococcus pyogenes or Group A strep, which causes strep throat, and Bordetella pertussis, the bacteria behind whooping cough. Molecular methods are also used for resistance testing, like carbamate nemase resistance, which helps identify antibiotic-resistant bacteria more swiftly. In addition, tests for Streptococcus agalactui, known as Group B strep, Chlamydia trachomatis, and Neisseria gonorrhoeae have all shifted from traditional culture-based methods to molecular diagnostics. Overall, these advancements highlight how molecular techniques streamline our diagnostic process, making it faster, more sensitive, and more comprehensive, truly transforming the landscape of microbiology testing in labs today. Most laboratories have shifted from traditional viral cultures to molecular methods for detecting viruses. This transition has improved the speed and accuracy of diagnosis. Some common examples, in addition to the ones we've already discussed, include influenza, respiratory syncytial virus, or RSV, herpes simplex virus types 1 and 2, varicella zoster virus, cytomegalovirus, and enterovirus. These molecular techniques allow for more rapid identification, which is crucial for timely patient management. It's also important to note that for new and emerging viruses like SARS-CoV-2 and H1N1, or swine flu, molecular methods are now available as well. This has been a game changer, especially during outbreaks, because it enables us to detect these viruses quickly and accurately, helping inform public health responses and treatment decisions. Overall, the shift to molecular diagnostics reflects the ongoing advancement in laboratory medicine, aligning with our goal of providing faster, more reliable results to improve patient care and control infectious diseases more effectively. Let's now consider the relevance of conventional techniques in clinical microbiology from the Indian perspective. Currently, India has over 1 million laboratories, both small and large, operating across the country. Despite this vast network, when the COVID-19 pandemic hit, only 52 labs were initially capable of performing emergency molecular testing. This highlights a significant gap in our capacity for rapid molecular diagnostics. Over the next 2 years, that number increased to over 3,300 labs, which is a commendable growth. However, this also reveals that, as a nation, we are still not fully prepared to rely solely on molecular testing for infectious disease diagnosis. Therefore, while we work towards scaling up molecular techniques, it's crucial to remember the importance of conventional microbiology methods. These traditional techniques remain fundamental and reliable in our diagnostic arsenal. Revisiting these methods is essential, especially in the context of integrating new technologies. They continue to play a vital role in our overall diagnostic strategy, ensuring we provide timely and accurate diagnoses even as we adopt advanced molecular tools. This balance between conventional and molecular methods is key to strengthening our infectious disease management in India. So, looking ahead, I believe the future of clinical microbiology will be shaped by continued technological advancements. We'll see molecular diagnostics expanding exponentially, which is incredibly exciting because it means faster, more accurate results that can really improve patient care. At the same time, I don't think traditional, conventional methods will disappear overnight. In fact, they will likely remain relevant for quite a long time, maybe even forever. These methods provide foundational insights and are often more accessible in certain settings. So, it's not about one replacing the other, but rather how they can complement each other to create a more robust and effective diagnostic landscape. This ongoing evolution aligns with our overall goal of enhancing diagnostics and improving patient outcomes through innovation and tradition working hand in hand. This slide doesn't contain enough information. That's why we're moving to the next slide.